Okay, you can see my screen just fine. Super. Okay, well, thank you very much, George and Tara, for that really kind introduction. And I'd like to say hi to all of those that are present here, some names that I can see and I can recognize. I wish we had more time to talk and visit. Um, okay, so I'll just begin. I don't think I need to convince anyone here the importance of skeletal muscle to whole body health. I think we all recognize that muscle makes up about 40% of body weight in a healthy individual. We know that it's not only important for movement, but it's required for ventilation, thermoregulation, as well as glucose disposal. So unfortunately, when we lose skeletal muscle mass, of course, this can lead to metabolic disease, disabilities, and early mortality. And this is exceptionally prevalent in our older adults. So if you just look at the dotted line, I'm sure most of you are familiar with this graph. It shows the normal progression of muscle loss after the age of 40. And it's estimated we lose about 1 to 2% of muscle mass every year after the age of 40, 50. Now we can make good decisions about exercise and nutrition. We can improve this progression, but we can't fully mitigate that loss. Now, unfortunately, many individuals will follow this lower trajectory of muscle loss. So we know that as we age, surgeries, illnesses, and injuries become relatively common. And with each one of these events that might require a period of bed rest or single limb immobilization, this can lead to the exacerbation of muscle loss that's just never fully recovered. And unfortunately, this can lead to the early onset of disability, at age 65 versus 80. Well, why do we care? Well, we all care because we're all going to grow old if we're lucky. And we all certainly care about quality of life. We want to live the best life that we can as long as possible. But practical concerns, of course, include the fact that we spend, at least in the US, a significant portion of our expenditures on taking care of those with disabilities. But more concerning is the fact that we recognize now that there's a shortage of home healthcare workers, which is known as the caregiver gap. Simply stated, there's just not enough healthcare workers to take care of those with disabilities. So we really need to, address this critical issue. Now, I think everyone would agree that resistance exercise is the most effective, most natural approach to rehabilitate muscle after disuse, but we can certainly appreciate some of the limitations for older adults in fully engaging in an exercise program. So as we age, we have impairments in mobility and balance, but there's also a lower threshold for pain. And even if we can engage fully in a rehabilitation program, there remains this challenge. There's this decreased capacity for regrowth that occurs after a period of disuse. And we don't fully understand the molecular mechanisms responsible, but it's been reported in many studies now, both in humans and in our animal models. But I think that this is really best exemplified by this paper here that was published by Charlotte Sueda, who's at the University of Copenhagen. This was published back in 2013, where she recruited both young and older adults to the laboratory, and she subjected them to unilateral casting. So she casted one of the limbs for 2 weeks, had them return to the lab, remove the cast, and then had them engage in unilateral strength training for 4 weeks. No big surprises here. Of course, there was a significant decrease in the size of the type 2 fibers, both in the young and the old. But here we can appreciate the fact that whereas the young fully recovered, there was no recovery in the old. So these observations have really stimulated some interesting questions in my lab over the last few years. First, what's mechanistically, what's responsible for this lack of regrowth after a period of disuse? And I want to clarify again that we're not interested in necessarily understanding the mechanisms for the loss of muscle during disuse. We're really more focused on trying to understand why there's this lack of regrowth after that period. And if we can address these mechanisms, identify them to some extent, then we could perhaps develop more targeted therapies to boost recovery, particularly in older adults. Okay, so what do we currently understand about the mechanisms responsible for this lack of recovery? Well, we've known since the early 60s as well as some work by Scott Powers, Lee G, David Hood, who's on the call, and others, that there's a significant decrease in mitochondrial content and function that occurs during a period of disuse. And that just makes sense. You know, if we're not contracting the muscle, there's less need for energy, maybe there's less need for this organelle structure. But unfortunately, what happens is when we start to remobilize that affected limb, we're contracting the muscle again. This is going to cause significant stress to the remaining mitochondria in the muscle, a bioenergetic stress that is going to allow for the release of reactive oxygen species. So this would include the reactive superoxide anions, as well as the more stable hydrogen peroxide. And this enhancement of ROS leads to, very importantly, lipid peroxidation, which is simply the scavenging of electrons from the lipids within our membranes. So our membranes are going to be significantly impaired and compromised. There's going to be increased ubiquitination and either degradation or aggregation of critical muscle proteins, as well as DNA damage. Now, in the case of an acute bout of exercise and an acute increase in ROS, we know that this can lead to the activation of signaling pathways to enhance antioxidant synthesis to really help balance that ROS. But unfortunately, in this context, we know that for whatever reason, with this chronic high level of ROS, that this can impair some of the signaling pathways, leading to a lack of antioxidant synthesis. And I think this is an area of research that is highly understudied. And so I'm really going to focus here a little bit in my talk. But together, this enhancement of ROS and this lack of antioxidant synthesis can really be devastating to our muscle, leading to this decreased capacity to recover. So all this information is important. It is truly informative, but there's a limitation, and that's the fact that most of the studies that have been conducted so far have been done using whole muscle samples, particularly in our animal models. And unfortunately, we know very little, relatively little, about the contribution of other mononuclear cells that are found that are resident within skeletal muscle. So this would include, of course, our muscle stem cells or satellite cells found here. These are important, of course, for repair and regeneration of muscle in response to injury. We have our fibrodipogenic progenitor cells, or FAPs, which are found in the interstitium, and they're going to turn over the ECM, which is important for muscle health. We also have vascular support cells like pericytes and smooth muscle cells, and we have a wide variety of immune cells like macrophages. Now in my laboratory, we have a specific interest in pericytes, and we feel that this is a very understudied, underappreciated cell type. And if you're not familiar with this vascular support or mural cell, you can look at it here in this illustration on the right-hand side in green. It's identified typically by having a large cell body, and it's going to send out projections, multiple projections that are going to wrap around and directly contact the endothelial cells of the microvessels. So this would include our venules, arterioles, and capillaries. And because of that, close localization to the vessels, the pericytes are predominantly known for regulating vascular health, right? So we know that they can regulate angiogenesis, dilation constriction processes, as well as they can regulate permeability of substances across the endothelial membrane. But we know, because we've studied these cells for many years, that they can also serve as a primary mesenchymal, stromal, or secretory cell in muscle. And we know that because they do express CD146, which is an important MSC marker. And we know because we've demonstrated in other publications that they can synthesize and release a wide variety of beneficial factors. So proangiogenic factors, anti-inflammatory cytokines, and importantly, antioxidants. And so given the information that we already know about pericytes and their importance to muscle health, we were certainly interested in understanding, well, do pericytes maybe lose the capacity to release some of these beneficial factors after disuse? So to address this question, the first thing we did several years ago now was we developed a model of immobilization that could be used with mice. We worked with Christopher Adams, who's now at Mayo Clinic, to develop the staple-based model of immobilization, which you can see here. And this is very simple. We're just going to tether the bottom of the foot to the shin in full dorsiflexion, which causes significant shortening and atrophy of the tibialis anterior. And then at 14 days, so 2 weeks of immobilization, this is a unilateral model, right? So we're going to be immobilizing one leg and the other leg is healthy and mobile. So at 14 days, we remove the staple from that one limb. And then if we observe the muscle 14 days later, we still see significant deficits in terms of fiber size inside the muscle and vascular refraction. Those are all still present. So this is a really rigorous model of immobilization. So here, what we did was at 3 days following remobilization, so after removal of the staple, we dissected the TA muscles from the immobilized limb as well as the mobile limb. And we isolated nuclei from pooled samples such that we were able to collect about 8,000 nuclei per limb. And then once we had the nuclei, we completed the cDNA barcoding as well as the library construction sequencing, the normal 10x genomics workflow, worked with bioinformatics to identify then our cell clusters. Okay, so here we're able to identify in our UMAPs our myonuclei, which comprise most of our nuclei that were isolated. So this would be our mainly type 2B, type 2X, type 2A, another type 2B cluster as well. We were able to identify our muscle stem cells, or FAPs, as well as our pericytes of interest. Okay, so now there's not enough time to talk about the reaction of all these different cell types to disuse and remobilization. But I can tell you that they were all affected. They were all impacted by this procedure. But let's just focus on the pericytes. So here, what we found was that there were 120 genes that were differentially regulated. And specifically, we found that there was a significant decrease in 30 important RNAs that met a specific threshold for significance. And what we found by completing our classification analysis was that almost all of these were related to the class ECM receptor interaction. And looking more closely, we could see that most of these were transmembrane proteins. So they were found at the membrane. And this is the membrane of the pericyte, remember. Now, one really stood out to us as being potentially impactful, and that is LRP1, which is the LDL receptor-related protein 1. And the reason why LRP1 is important is because, very similar to what you can see here for LRP8, it's critically important for the transportation of selenium, mainly in the form of selenoprotein P, into a cell that is then therefore important for not only transcription, but translation of important antioxidants such as glutathione peroxidases, such as GPX4 and GPX1. And what's really important about GPX4 is that this particular GPX can uniquely directly reduce lipid hydroperoxides from the membrane. Okay, so no other antioxidant has that capacity. So if you are not synthesizing the LRP receptor, then you're not transporting that critical selenium, and you're then preventing the ability to manufacture this antioxidant, leading to a regulated cell death process known as ferroptosis. So it's a relatively new mechanism responsible for cell death. So there have been some papers published just in this last year. Henrietta van Praag has been one of the authors of some of these papers, where they show that GPX4 is a critical regulator of muscle mass in the context of disuse, as well as neuromuscular disease and aging. And so this was a potential target, this LRP1 GPX4 mechanism. Now, again, this will come up in just a minute, but LRP1 is also important for the manufacturing of GPX1, but just kind of keep in mind that GPX1 only can reduce hydrogen peroxide, the more stable ROS, and not these lipid hydroperoxides. Okay, so it's a little different in its capacity. Okay, so given some information then suggesting that, the pericytes may be deficient in the capacity to synthesize and release these important antioxidants. Yu-Fu Wu in the laboratory, who since graduated, he conducted an experiment where, again, at this critical period, 3 days post-remobilization, he isolated the pericytes from the muscle, the TA muscles, the pericytes from these TA muscles, and then isolated the RNA, and also then completed a targeted qPCR analysis of critical antioxidants, including GPX4. And you can see here that the pericytes that were isolated from the muscle were really not responsive to the enhancement of ROS, which we were able to verify. So there was no significant increase in antioxidant synthesis. And so we thought, well, maybe we just missed the time point. Maybe there was an enhancement of antioxidant synthesis at, I don't know, 1 day post remobilization. And so we thought, well, let's try something else. Maybe we could assess this in vitro. And so again, what Yu-Fu did was he isolated the pericytes, placed them in culture for a short period of time, less than 10 days. And then he added hydrogen peroxide at a level that was really considered physiological, 50 micromolar. And then again, we extracted the RNA and completed the targeted analysis. And what we found was that that there was, if you look at the orange bars, there was a nice response in terms of the ability for antioxidants to be synthesized in the healthy pericytes from the mobile leg, but there's really no response at all in the pericytes from the immobilized leg. And this was particularly true for GPX4. Okay, so that was really interesting, gave us a little bit more evidence, but this is gene transcription, and certainly we want a little more information about pericyte antioxidant function, the ability for these antioxidants to impact the muscle. And so what we did here was, again, this is Yu-Fu, and so after the immobilization period, what he decided to do was inject the pericytes from the healthy limb that we knew synthesized those antioxidants. But as well, he injected in a separate cohort of animals, he injected then the pericytes from the immobilized leg that we knew were deficient. And then 3 days later, he evaluated ROS in the muscle based on 8-OHDG and 4-H&E assays, as well as ubiquitination, a measure of protein breakdown. And so what he found, as hypothesized, was that when he injected the healthy pericytes from the mobile leg, there was a decrease in ROS, again, based on this assay, as well as 4-H&E, which is not shown here, but no change when he injected the pericytes from the immobilized leg. So they didn't have that capacity to reduce ROS. And then same for ubiquitination, nice decrease, suppression when we injected those healthy pericytes, but no response with the immobilized cells. Okay, so these results, these observations likely explain some of the data that we collected and we published back earlier in 2019, where we just simply hypothesized that these pericytes might be helpful in recovering muscle. And so what Michael Monroe did here, again, a former student, was that he collected limbs 14 days after remobilization, a much later time point, where we knew there were still these deficits. You can see here in the fiber CSA, as well as the capillary to fiber ratio and capillary density. But when he injected the pericytes versus a hydrogel control, you can see that there's nice recovery of the myofiber CSA, as well as recovery of this capillary to fiber ratio, and even some enhancement of the capillary density. Okay, so what we're doing right now is we are taking a deeper dive in vitro. And so this is work by a new postdoc in the lab, Anne Vigian, where she is collecting, again, these primary pericytes 3 days post-remobilization. She places them in culture. And then by mass spec, we're able to evaluate the levels of LRP1 and these critical glutathione peroxidases. And so based on protein levels, we can see that there is a significant drop in LRP1 and there's a significant drop in GPX1. And we believe that there's a decrease in GPX4, but it's in much lower concentration than GPX1. And so we're now confirming that decrease with standards. We need to make sure that what we're showing is real. So far, it looks like there is this defect in the ability for the pericytes to synthesize these important antioxidants. So here's an illustration from Onatera's reviews. I really like this one because it shows the pericytes next to the vessels impacting then the surrounding fibers. This is how I view pericyte fiber interactions as well. And so we're able to demonstrate so far some evidence that pericytes are negatively impacted by disuse and that they can lose this capacity to secrete antioxidants that might be really important for reducing ROS inside the muscle. But what about aged mice, right? So here we're able to demonstrate the ability for the pericytes to help with the recovery in younger animals, the young adult. But what about older animals? And so here's some data that we haven't published yet, just specifically looking at the impact of age on pericytes in the muscle. And so again, this is single cell, not single nuclei, but single cell data, again, not published, where we're comparing young and the old animals. We're able to find our muscle stem cells, which are decreased, which has been reported before. We find our FAPs, and then it looks like there's also potentially a decrease in pericyte quantity. And then a new graduate student in the lab has taken up this project, and she's been able to isolate the pericytes from the aged muscle, and she can see that there is an increase in senescence based on beta-gal stain. So here, the gene signature of this pericyte cluster indicated that these cells were becoming, in fact, senescent, even perhaps more so than what we were finding with the fibroblasts. We thought that was interesting, and so she was able to confirm that they are, in fact, highly senescent. So far, these results would suggest that pericytes, aged pericytes, are decreased perhaps in quantity and quality in muscle. And so we thought, well, this would, of course, provide nice justification for injecting these cells, these pericytes, and determining whether they could enhance recovery. And I can unfortunately tell you that they do not have that capacity. So this was data that we actually collected a long time ago, not published for good reason, where we injected our young, healthy pericytes into now 24-month-old males. This is a direct muscle injection. And then we evaluated the recovery 14 days later. And you can see there's really no recovery at all in terms of the myofiber CSA as well as capillary content function. So this isn't terribly surprising because we've conducted a lot of these different cell injection replacement studies in the past, and they really don't have really high regenerative capacity. And we believe it's because they are, encountering this collagen-enriched hostile environment. And so in that environment, they are really dysfunctional. And so this was certainly a challenge that we needed to think about and perhaps and really address and get around. So it was about this time, about 3 or 4 years ago, that we started to read some papers suggesting that mesenchymal stem cells, stromal cells, can release small extracellular vesicles that can reduce oxidative stress, inflammation, and fibrosis. And I was intrigued. And I assume everyone here is familiar with EVs, but very quickly we know that these are very small lipid membrane-bound vesicles that are released by nearly all cells in response to a variety of stimuli. The small EVs or exosomes range in size less than 200 nanometers, and they're critically important for intercellular communication. Because they can transfer molecular cargo, mainly MIRs, but also proteins from a donating cell to a nearby or distal cell. Okay, so these are little packages of cells, right, that are shared. And there's some thought now that these MSC-derived EVs are going to, in the future, be preferred over cell therapies because number one, they can be stored, at least for a short time. So they're not live. Like you can't freeze, a live cell and have it come back and be regenerative. That's really problematic. But EVs can be stored, again, for a relatively decent period of time, about 2 months, 2 to 3 months max. They're less immunogenic, we believe, although there's still some question in the field whether or not EVs are rejected once they're injected into, recipient animal. And then finally, they are highly stable in biological fluids and tissues, less opportunity for rejection. Okay, so given this information, then we decided to obtain our pericytes, again, from healthy muscle now. We plated them in culture, as you can see here, and then this is a short period of time until they were confluent. And then we either treated them with just media, or again, we primed them with hydrogen peroxide at the physiological level that allows them to synthesize antioxidants. We then collected the conditioned media 48 hours later. We condensed the media with an ultrafiltration procedure. And then we used size exclusion chromatography, SEC, as our method for enriching our EV within the media. After one more ultrafiltration step, we had our EVs for injection. So just quickly, we were able to thoroughly characterize the EVs that we collected. So here, using NTA, we're able to identify the EVs predominantly in fractions 5 and 6, but we're collecting 4 through 6 as our EV fraction. And we're able to verify that these EVs expressed a wide variety of EV markers. So this would include our ALEX, CD, 63, 81, and 9. In terms of size, the size was around 130 nanometers, no significant difference with the priming. Okay, so here again, this was Yufu's work, and he used 4 and 24-month-old animals. After the immobilization, he injected these pericyte-derived EVs, either the unprimed or primed condition. He evaluated ROS at 3 days, as well as the fiber CSA 14 days later. And so what he found was that in the younger adult animals, there was, in fact, a significant reduction in the muscle pore H&E when he injected those pericyte-derived EVs after that priming condition. So that was really nice to see. No change in the older adults. You can see that the data was quite variable. But regardless, what we found was that there was a significant, pretty robust increase in the myofiber CSA. So you can see that both for the young animals as well as for the aged animals as well. And importantly, if you look at the fiber size distribution, you can see that the fibers that we're predominantly affecting are those ranging greater than 3,000 microns. So the larger size fibers within the muscle, you can see that we're really recovering those both in the young as well as in the aged animals. That was good to see. Okay, so clearly we're interested in understanding, well, what in the world was in these EVs that might be driving some of these beneficial effects? And so we have a collaborator, Jonathan Sweetler, who's in the Department of Chemistry here at the U of I, and he has expertise in assessing or identifying low abundant proteins in samples using mass spec. And so he was able to analyze our samples. He found that there were 802 unique proteins identified in the non-primed, unprimed, and primed condition. There was significant overlap with data from Exocarta, which reports any EV content, so from a variety of different papers in the field. Importantly, he found that there were 78 different proteins that were differentially regulated between the primed and unprimed condition. And so here you can see that many of the top upregulated proteins were ECM-related proteins, so proteins found in the extracellular matrix. Contraxin 3 has been found to be secreted by pericytes, and it can serve both an antioxidant and anti-inflammatory role. But we didn't, in terms of glutathione peroxidase, it didn't seem to rise to the top until we performed a protein class analysis. And here the top protein protein class was metabolite interconversion enzymes, specifically our oxidoreductases or antioxidants. And so here's a list of all the different antioxidants that we found enriched in the EVs. And this included glutathione peroxidase 1 or GPX1. Interestingly, we also found that there was enrichment of LRP1 as well. And we are still verifying glutathione peroxidase 4, which is again found in a much lower level. So based on that information and some other data from other experiments, we decided that we wanted to maybe pursue an experiment where we created our own nanoparticles. So artificial EVs, if you will. And so we have a collaborator here, longtime collaborator, Jun Kang, who's in the Department of Chemical Engineering, who has expertise in developing different substrates and polymers for regenerative purposes. And so here he decided he wanted to use an FDA-approved PLGA hydrolonic acid nanoparticle. So material to build this nanoparticle. And we decided to take a very unique approach whereby we could encapsulate not just our protein of interest, like our GPXs, but also magnesium oxide, or similar, we sometimes use as catalase. And the reason why you package the catalase is because what can happen is that once these nanoparticles that possess the magnesium oxide or the catalase enter the microenvironment that is ROS enriched, the ROS is going to enter directly the nanoparticle, and then it's going to decompose the ROS and generate oxygen gas, a pressure that's going to slowly allow for the release of our protein of interest. And this is really important because if you know about antioxidants, you might understand that there has to be a critical balance, right, of antioxidants within a tissue. If you have too much antioxidant, that can cause reductive stress. And so what we wanted was the release of antioxidants in an appropriate level. And so this, redox sensitive instrument gave us that capacity. So in this first experiment here that we've just completed, we were able to generate GPX1 loaded artificial EVs. And so we injected them here again at 14 days, evaluated the muscle at 3 days. And we also had a lipid nanoparticle control that was devoid of GPX1. And what we found was, no changes in muscle so far, which is not surprising because remember, GPX1 isn't able to reduce those critical hydroperoxides in the membrane. They're mainly targeting hydrogen peroxide. And we know that GPX1 is more critical for the vascular health. And so we we evaluated the capillary to fiber ratio, and you can see that as expected, there's a drop with the immobilization procedure. But when we inject, then the GPX1 nanoparticles, there's nice recovery, and you can see the difference here as well in some of our images. So it had a very profound effect in recapturing a lot of those capillaries. So we're really excited about taking this approach to therapy. Okay, so we can add then to our summary that the pericyte-derived EVs improve recovery of muscle, specifically the fiber CSA, fiber size after disuse in H mice. So that was really nice to see. And these GPX1 loaded nanoparticles provide this robust capacity to improve capillary regrowth. Okay, so what are we doing now? Well, after we verify in vitro that the pericytes are in fact, producing GPX4 as well as GPX1, and that this is deficient after this disuse period, we want to perform a variety of in vivo experiments. So we've been working with a pericyte-specific cray mouse. So this is PDGF receptor beta cray. So this is going to allow for the conditional expression of the cray recombinase. And we've already combined these with a reporter mouse, MT-MG animals. And you can see some of the images here from our multimodal microscopy procedure. So in green in the GFP, you can see the pericytes in close localization to the vessels. And we can even start to appreciate perhaps some small, perhaps particles then inside the vessel, but we'll see. But critically, we're wondering, can we use these animals to deplete pericytes specifically in muscle and determine the impact on recovery? So we've already set up breeder pairs with the PGF-Cray by DTA so that we can, upon injection with the tamoxifen, we can deplete those pericytes and determine the impact on recovery. If that's true, that those pericytes are really critical, they provide a significant contribution to recovery. The next step, of course, is to deplete LRP1 in the pericyte. So we were able to, again, take this Cray mouse, combine it with the LRP1 flox mouse, and determine the impact then specifically of GPX4 and GPX1. And then finally, we can either overexpress GPX4 using the same animal model, or we're just going ahead right now, creating the GPX4 nanoparticles and determining the impact on muscle health. So with that, I'd just like to thank all of the students that completed the work that was shown here, as well as my collaborators and the funding that allowed this work to be possible. So thanks so much for your attention. I'd be happy to take any questions.
Oh, thanks, Marni. That was a really fascinating talk. So much exciting projects that you're working on. For those of you who have questions, you can either raise your hand and put your camera on and ask, or you can put a question in the chat if you prefer that. To begin, while people are still composing their questions, for the experiment where you were injecting the pericytes into the aged animals and you didn't see any improvement, was it because the pericytes didn't survive when you did the injections, or was it something about just the environment didn't enable them to function in the same way?
Yeah, so we didn't directly look at viability. So we didn't try to complete that assessment, but we have published papers. Mike Delisio, I see, is on the call here, and he was able to demonstrate that with similar cells, if you place them in a collagen-enriched environment, that they start, their paracrine secretion is different, right? And so it's they're secreting less beneficial factors, and they're just not the same cell, right? So my guess would be that those pericytes that we're injecting are really not the same pericytes, right, that we found when we injected into the younger mice in that healthy environment. They're just not, they're dysfunctional. Mm-hmm. Yeah, in their capacity. Mm-hmm.
How do you work out the amount of cells or vesicles to inject into a muscle? I would imagine that you couldn't just target a single area. You would want to ensure it's distributed well enough to see a functional effect.
Yeah, so we have these conversations all the time. Where do we start with the concentration, right? Is that your question? Like, what's the quantity you inject? So we based it on other papers, right, where they're using MSC-derived EVs. And so that was really our starting point. Is there an optimal amount? Probably. But we just chose, a concentration just based on the literature, and we had good results. And so that was wonderful to see. But maybe there's some even more room for improvement, or it's just really hard to say. The work is really challenging, like any experiment, right? And so you're trying to capture, it's a long procedure to get the EVs, right, in the first place. And so, these aren't easy experiments. We can't do a lot of these optimization experiments. So sometimes we just have to guess and hope for the best and, you know, cross your fingers. And we just lucked out.
Yeah, for sure.
Mhm.
Any questions from the audience?
George has his hand up.
Yeah.
Okay.
Hey, Dr. Boppart, thank you for your talk. It was a very good talk. Just I have like two questions. In terms of the model of the unilateral immobilization, do you think the results are gonna be different if you compare the immobilized leg parasite with a completely different control mouse, other than maybe overloading the other leg of the mouse by just like immobilizing one of them and overload the other one?
Yeah, we get that question often. So from reviewers, you know, is the mobile control altered in some way, right, from the same mouse? And we did initially a variety of experiments to test whether or not the mobile leg was different from a complete control. And we found very little difference. And so we decided to continue with the unilateral model because it does provide, you know, an internal control. So, you know, your data is a little bit tighter. Yeah, so we did test that and we decided to continue this way. It saves a lot of money too,
Oh, yeah.
with ordering mice as well. But yeah, good question. Great.
Sure. Sorry, I have another question.
Yeah.
You know that parasites are not like a homogeneous population in terms of like different subtypes that were reported previously. So do you think if you choose like another population of parasite that might be like CD 146 negative and maybe NG2 positive, do you think we're gonna see like, did you test any of those to see if there is like different results, or it just like maybe like something you think should be like studied?
Yeah, so we published a paper 2019 where we compared NG2 positive parasites and their capacity versus CD146. We didn't test PGF receptor beta yet, but those two were the primary. And we found that the CD146 had greater capacity so for recovery. And so that's why we've continued with CD146.
Sure, thank you.
Okay, there's a few questions in the chat. George Pavis asks, thank you, Dr. Boppart, for the talk, a very impressive and comprehensive series of studies. I wondered to what extent these mechanisms might track to humans. It appears that immobilization is a more aggressive intervention in rodents than humans, greater rates of muscle mass loss, and upregulation of breakdown in rodents. So I wondered whether this parasite inflammation axis might reflect the nature of immobilization specifically in rodents.
Okay, well, yeah, so we believe that the parasites are impacting ROS. We really haven't tested their capacity for addressing inflammation inside the muscle. But I think I'm thinking that you believe that maybe inflammation is part of, you know, some of the deficits. It's responsible for some of those deficits. But we haven't tested that. And so, you know, we have to start somewhere, right? I mean, it's important that if you're developing a therapy that you show that it's beneficial in an animal model first. I will tell you that we have started to collect EVs from humans for injection in the mouse model still, but at least getting a little bit closer to understanding, you know, what is the ability for these EVs still to have this capacity, whether they're derived from mouse or from human. And so hopefully getting a little bit closer to making this a little bit more translational. But again, you just need to start somewhere. So this is it. I will also mention we've developed the hind limb suspension model as well. So we are looking at a different model that's a little less aggressive than this staple-based model. And we should have those results soon.
And another question from the chat, Damien Callahan. It seemed as though the response to Perry's site-loaded EVs were related to the extracellular matrix. Did the tissue become fibrotic? And was muscle recovery dominated by contractile or non-contractile tissue?
Yeah, so we did assess the ECM turnover with CHP assay as well as collagen assays. It's in the paper that was just published this year. And it looked like there was nice capacity for the EVs to assist with the turnover. So it looks like there is a little bit of fibrosis, and it looks like there's an increase in the turnover of some of that collagen. So that's an excellent question. How do the EVs do that? I don't know necessarily yet, but that is part of the recovery process. So downloading the paper now, great. It's in there, yeah. Mhm.
Any other questions for Dr. Boppart?
think those-
Damien, you have a question? Or was that from what you had?
No, that was actually on purpose. Sorry. And sorry to take a minute there. I figured it'd be a little less time than typing out. But following up on that, given all the recent evidence suggesting that satellite cell activation is required and integration is required for muscle hypertrophic responses, and that myonuclei, at least in mice, are not post-mitotic, do you see a potential for interaction between loaded parasites and activation of quiescent satellite cells, or furthering division in existing myonuclei to kind of support this recovery? And layer it on top of that, do you see this recovery from disuse or a lack of atrophy during disuse to be similar mechanistically to trophic hypertrophy in response to loading?
So I
So
believe
there was 9 questions. I'm
yeah,
sorry.
I know. I'm trying to tackle them all at once. So Esther DuPont Verstegen, she published a paper long ago, I want to say 2008, where she depleted satellite cells from muscle and then did the disuse reload, and she found no difference at all. So in the context of disuse recovery, satellite cells probably don't play an important role. It was a nice paper, and so right now we're doing a cell chat analysis to look at the communication between the different cell types within the context of our single nuclei sequencing results, and that'll give us some hint as to whether or not these cells are interacting and communicating with one another. But that's just predictive, right? We're not directly analyzing that relationship. I'm less interested in satellite cells because, and it's not, I think we all know satellite cells are important for muscle recovery after injury, right? But I don't think disuse is necessarily an injury in the same way. So I think that other cell types are really have a greater contribution to the recovery process. But that's just my opinion.
Yeah, thanks. Really interesting work and a great talk, at least for the portions I was able to attend. I was running late from another meeting. But yeah, I guess my question was sort of tangentially getting at the point that they're different processes. And your work is really, I think, is highlighting the really important elements that are specific to disuse related atrophy, as opposed to just being a different slider on the continuum of how big your muscle is going to be.
Exactly. That's what I tried to emphasize in the beginning too, is that, in this context, there's this enhancement of ROS that's really unique characteristic of the recovery period. That you may or may not see in the context of a healthy, muscle contraction, acute muscle contraction context. Is very different. Mm-hmm.
Thanks for the talk and for entertaining my questions.
Yeah, thank you.
did you do experiments as well, just collecting the conditioned media from the cultured pericytes and injecting that into the muscle? Or is that just a silly idea?
No. So we are doing, so we have different projects going on in the lab right now with EVs. And what we have learned since the time that this work was completed is that it's really important to inject the fractions from the SEC that are devoid of EVs as a nice control. And that's been really informative. And so far we find differential results when we inject the EVs versus the fractions that have the protein without the EVs. So in the EV world, right, it's really important to have as many controls as possible. You need to have the control with the depletion of the EV. So yeah, that's part of our current projects. Yeah. Thanks for asking. That's a great question.
Okay, well, if there's no further questions, then I'll call a close to the seminar and stop the recording. If anyone does remaining have questions, just stay on, and I'm sure we can stick around for a little bit and continue to chat if you still have something to ask.